Hormones exert their effects through four distinct classes of receptors. The first type of receptor is intracellular and resides in the nucleus. These are the steroid hormone receptors with specificity for different steroid hormones. The second type of receptors resides in the membrane, and an example of that is the nicotinic cholinergic receptor activation by acetylcholine, which allows this channel to open. The third type of membrane receptors is the receptor enzyme, and an example of that is the insulin receptor, which when activated by insulin will activate the tyrosine kinase activity of the receptor. The fourth type of membrane receptors is G-protein coupled receptors, which reside in the membrane, and they constitute a large family of receptors. An example of these receptors are the beta-adrenergic receptor, the glucagon receptor, and the alpha-1 adrenergic receptor. All of these G-protein coupled receptors transduce their intracellular effects through activation of specific G proteins. Importantly, the activation of these G proteins results in the activation of a specific effector enzyme. The activation of these effector enzymes results in the production of unique and specific intracellular second messengers inside the cytoplasm.
This receptor is called the cortisol receptor. They bind, recognize each other, and form this complex. This is the active form. Each one separately or alone is not active. Together, they're active. Now, they translocate from the cytoplasm into the nucleus, bind to an enhancer region in a specific gene that activates the promoter region of that gene and increases transcription. So this is what you call upregulation or induction of genes. More messenger RNA is translated to more protein. So when you activate the insulin receptor, you activate the tyrosine kinase activity of that receptor. I want you to appreciate that in that beta subunit shown here, this is again a polypeptide. There are several tyrosine residues, part of that polypeptide. They get phosphorylated by the intrinsic kinase activity of the receptor. That's called autophosphorylation, meaning the receptor phosphorylates its own subunits on specific tyrosine residues. These aren't loose tyrosines. They're part of the receptor itself, of the polypeptide making up the beta subunits. Very important. So that is a sign that the receptor has been activated. One of the key things about G-protein coupled receptors is when a G-protein coupled receptor is activated, it will invariably produce an intracellular second messenger. Okay? Cyclic AMP is an example of a second messenger. Before receptor activation, this molecule would have been absent inside the cytoplasm. After receptor activation, this molecule concentration goes up. And that's why we call that a second messenger. Second important feature about second messengers is that they are typically small organic molecules. Okay? They are not proteins in nature. They are not peptides. They are typically organic small molecules. Here they are. Here are the examples. Cyclic AMP is one, IP3, inositol trisphosphate, diacylglycerol, calcium, cyclic GMP, nitric oxide. Second messengers are typically small molecules that are not present during the resting state of a cell.
Activation of either the beta adrenergic receptor by epinephrine or the glucagon receptor by glucagon activates GS-alpha, which in turn activates adenylate cyclase that produces the second messenger cyclic AMP. G-protein coupled receptors are highly specific. Only epinephrine can activate the beta adrenergic receptor, and only glucagon can activate the glucagon receptor. Activation of the alpha-1 adrenergic receptor by epinephrine results in the activation of the trimeric G protein designated GQ. GQ dissociates to produce GQ alpha bound to GTP, which then can activate phospholipase C. Phospholipase C will hydrolyze PIP2 in the membrane to produce diacylglycerol DG in the membrane and IP3 in the cytoplasm. IP3 moves to the ER and releases calcium from calcium stores in the ER. Ultimately, 3-second messengers are produced in response to receptor activation by epinephrine. Activation of the beta-1 adrenergic receptor by epinephrine or norepinephrine activates GS-alpha, which activates adenylate cyclase and produces cyclic AMP as a second messenger. Activation of the glucagon receptor by glucagon activates GS-alpha and activates adenylate cyclase to produce cyclic AMP as a second messenger. Activation of the alpha-1 adrenergic receptor by epinephrine or norepinephrine activates GQ-alpha to activate phospholipase C and produce IP3DG and calcium as second messengers. If you like this video, please subscribe. Thank you.